After spending the last 24,000 years in frozen hibernation alone, Rotifer awoke. Scientists hope that further research on this multicellular animal could lead to better cryopreserving of human cells, tissues, and organs. Rotifers are insect-like aquatic animals that prefer freshwater environments and moist soils. These complex organisms are not as charismatic as another tiny animal, the tardigrade, but are also known for their extreme survival skills because they can withstand dehydration, freezing temperatures, starvation, and hypoxic levels. Scientists have demonstrated that these creatures are even tougher than previously thought by reviving the 24,000-year-old rotifer found in the Siberian permafrost. The new findings were published today in Current Biology. It's an impressive feat for microbes, but the record is probably not. In 2018, Russian scientists claimed to have revived nematodes drawn from permafrost deposits at least 42,000 years ago. Previous rotifer recovery records range from 6 to 10 years and include specimens found in Antarctic soil and glaciers. Tardigrades have a record of 30 years, but given their track record, I secretly suspect they can withstand even longer hibernation. The credit for the new discovery lies in the Institute for Cryobiology of Soil in Russia. The laboratory uses a drill to dig samples from the Siberian permafrost. The resurrected rotifer was found at a depth of 11.5 feet, or 3.5 meters, near the Alizaya River in northeastern Siberia. The team used a special extraction method to prevent the contamination of adjacent layers and, at the same time, eliminate natural penetration from the upper layers. The late Pleistocene layer holding rotifer specimens was radiocarbon dated about 24,000 years ago. This was about 12,500 years before the end of the latest ice age. Scientists returned to the lab to revive the rotifer and even produce several secondary rotifer cultures from the same individual. Rotifers reproduce asexually in a process known as parthenogenesis, increase. The reported resurrection, according to the treatise, consists of the longest reported case of rotifer survival in the frozen state. Genetic analysis of the specimen confirmed that it belonged to the genus Adinetta and was fairly well compared to live samples supplied from Belgium. To learn more about rotifers and how to make this trick successful, the team randomly selected 144 unrelated rotifer specimens and froze them at 5 degrees Fahrenheit or -15 degrees Celsius for a week. Although limited in scope, this experiment showed that ancient Adinetta specimens were significantly less freeze-tolerant than modern species. The point is that multicellular organisms can be frozen and preserved for thousands of years and then revived. This is the dream of many fiction writers, says Malibin. Of course, the more complex the organism, the harder it is to freeze and store it alive. For mammals, this is currently not possible. Still, the transition from unicellular organisms to organisms with intestines and brains is. It's a microscopic but big step forward. For some reason, the Adina rotifer can fight the formation of ice crystals, the deadly enemy of the freezing process. Ice crystals are like small knives that destroy the integrity of cells and organs. According to the treatise, rotifers probably have the biochemical mechanism of organ and cell shields needed to withstand low temperatures. A better understanding of this rotifer's defense mechanism can greatly help improve cryopreservation techniques for preserving cells, organs, and tissues at low temperatures. More speculatively, these insights could even advance the early areas of cryonics in which dead humans are stored at liquid nitrogen temperature. The resurrection of these frozen people may never happen, but elastic rotifers at least give us hope. Early studies by other groups have shown that rotifers can survive up to 10 years when frozen. In a new study, Russian researchers used radiocarbon dating to recover creatures from the permafrost layer, ground that is frozen all year round except for a thin layer near the surface, about 24,000 years ago. I decided that it was. This study was published in the journal Current Biology on Monday. This is not the first time ancient life has been revived from a permanently frozen habitat. Antarctic moss stems successfully regrow from a 1,000-year-old sample that had been covered in ice for about 400 years, and living campion flowers were preserved by Arctic squirrels in 32,000 years, regenerated from seed tissue that appears to have been preserved, aged permafrost. A simple worm, called a nematode, has revived from 2 permafrost layers in northeastern Siberia with sediments more than 30,000 years old. Long dead but well-preserved mammals, such as the extinct cave bears and mammoths, have also been excavated from permafrost layers that have melted in several places as a result of the climate crisis. Malibin said it was very unlikely that he would be able to survive this frozen. The point is that multicellular organisms can be frozen and preserved for thousands of years and then revived. This is the dream of many fiction writers, Malibin said in a statement. Of course, the more complex the organism, the harder it is to freeze and store it alive. For mammals, it is currently impossible. However, the transition from unicellular organisms to organisms with intestines and brains is not possible. It's a microscopic but big step forward. When the rotifers thawed, the creatures were able to breed, the study said. Small invertebrates were also able to feed. Researchers frozen and thawed modern rotifers living in permafrost to understand how the creatures survived in the animation of hanging in the permafrost. They found that the creatures could withstand the formation of ice crystals while slowly freezing. Not all rotifers survived the freezing process, but this study suggests that organisms have some mechanism that can protect cells and organs from harm at very low temperatures. Researchers have previously discovered that modern rotifers freeze at -4 degrees Fahrenheit or -20 degrees Celsius and can resurrect in up to 10 years. Today, scientists have revived rotifers frozen in ancient Siberian permafrost in the second half of the Pleistocene, 2.6 million to about 11,700 years ago. When thawed, these ancient rotifers began asexual reproduction through parthenogenesis and cloned their genetic replicas. So, that's all for today's video. Thanks for watching the video. If you enjoyed it, make sure you hit like and subscribe to our channel. Let us know your thoughts in the comment section.